Okay, so I'll just skip to this slide. So I'm picturing here what happens to a healthy heart following what's commonly termed a heart attack, or more technically, a myocardial infarction, where we have massive death of cardiac tissue, and in place of this cardiac tissue, there's non-contractile scar that's formed, and unfortunately, in adults, tissue regeneration is not possible. And as such, ischemic heart disease remains one of the leading causes of death worldwide. So one of the approaches that many labs have taken is they've started to use stem cell-derived cardiomyocytes. And in pig models of heart attacks, we've seen that transplanting these stem cell-derived cardiomyocytes are able to stably engraft the infarcted pig heart and result in tissue regeneration. However, there's many challenges that remain with these cell-based therapy approaches. For example, there's massive cell death upon injection. There's 1 billion cells that's needed for one pig, and as a result, there's poor electrical integration, leading to deadly arrhythmias, as well as partial remuscularization. And what I'd like to point out is really the obvious, that the healthy myocardium is comprised not just of cardiomyocytes, but also a variety of immune and stromal cells that are required for normal cardiac function. And so this really raises the question of whether other support cells directly included could help with tissue regeneration. And as an immunologist, I'm interested in macrophages. Specifically, we have these live-1 positive, TIMD4 positive primitive macrophages that are derived from the yolk sac and fetal monocyte precursors during early embryonic development. Now, what's interesting about neonates is that cardiac regeneration actually is possible after injury. And from an immunology perspective, what's really interesting to us is that these primitive embryonic derived macrophages are required for the regenerative capacity of the neonatal heart. These macrophages persist into adulthood, where they're required for adaptive cardiomyocyte growth during hypertension, for repair following myocardial infarction, as well as several homeostatic functions such as electrical conduction and cardiomyocyte mitochondrial homeostasis. And more recently, we found correlates of this live-1 positive embryonic macrophages using human single-cell RNA sequencing of cardiac tissue. Now, while we appreciate that this era of cardioimmunology is expanding and we're learning more and more about the vital roles of macrophages, the major gap that remains is that we really don't know whether any of these findings in murine mouse studies are translatable to human. And so we don't know anything about the role of human macrophages in cardiac function. And this is largely due to gap number 2, which is that bioengineered cardiac tissues have focused solely on cardiomyocytes and stromal cells, and immune cells have never been incorporated despite being present in the native human heart. So the question that my PhD focuses on is, what is the role of human macrophages in human cardiac function? And to assess this, we collaborate with Dr. Gordon Keller's lab at U of T, and we take human embryonic stem cells, differentiate them through a stream that resembles primitive hematopoiesis to generate HESC-derived macrophages that resemble those that are regenerative in mice and neonates. We also use a human heart on a chip platform termed the BioWire, developed by Dr. Melissa Radisik's lab, also at U of T. And this system is comprised of cardiomyocytes and fibroblasts with or without macrophages, embedded in a matrigel collagen hydrogel, and it's seated within this little microwell in a 96-well plate. And over a week, it starts to compact and it forms this 3D cylindrical tissue that hangs on either side to a wire. The video would be beating to show you that we're able to really make these mini hearts within the lab. And what's really cool is that we can then assess the function of these human cardiac tissues and the role of macrophages in their function. And what we found is that incorporating macrophages are actually functionally active. So we see increased contractile force, improved relaxation, as well as improved calcium cycling, which together told us that macrophages improve a broad range of electromechanical properties in human cardiac microtissues. So we were then interested in assessing what aspect of cardiac function macrophages improve or alter within these tissues. So we performed proteomics using mass spectrometry of whole biowire tissues, both early and late following making these tissues, with or without macrophages. And while we made many findings with these data, I'd like to point out the most interesting which was that when we incorporate macrophages within tissues, we see upregulation of a broad range of contractile machinery proteins, as well as a shift from fetal to adult isoforms of contractile machinery, which together tells us that there's structural maturation of cardiomyocytes and biowires with macrophages, which is really a key finding because we know that maturation of cardiomyocytes is a struggle within the field. So at this point, we pose the question of whether macrophages alter metabolic pathways. Given that the heart is highly metabolic demanding and the function of the heart depends on large stores of ATP, we wondered whether macrophages may skew the pathways that cardiomyocytes use. And so to assess this, we performed cytometry by time of flight, or mass cytometry, using a panel that we created of 36 either cell type specific or metabolic pathway specific proteins. And so here what we found is that we had a variety of the cell types within our tissue, including cardiomyocytes, macrophages, and fibroblasts. But our major finding here, the most striking difference, was really this population of dead cells, or rather a population of dead cells or debris that had high expression of our dead marker, expressed DNA, and the only markers that they expressed were those that were localized to the mitochondria, including ATP5, which is a mitochondrial localized ATP synthase, as well as citrate synthase. We confirmed this using immunofluorescence, where we saw the accumulation of this punctate ATP5 positive mitochondrial debris within control microtissues, but not those with macrophages. And when we went back to our proteomics data, we actually saw that when we assess mitochondrial localized proteins, we see downregulation of those from a variety of pathways, including ATP synthases, mitochondrial ribosomal proteins, as well as structural proteins. And when we looked within the Krebs cycle, we saw that there are many isoforms of Krebs cycle proteins, including those that are localized to the cytosol versus those that are localized to the mitochondria. And what was interesting here is that the cytosolic localized mitochondrial Krebs cycle proteins were upregulated, whereas those that were localized to the mitochondria were downregulated. And so what this tells us is that there's reduced mitochondrial localized proteins, even within the same metabolic pathway. Now, despite all of these findings, we actually observed that within our whole microtissues, those with macrophages had higher ATP content. And so despite this reduction in mitochondria, we really attributed this to macrophages being able to reduce the accumulation of mitochondrial debris, but the overall metabolic output is increased. We then looked within our live cardiomyocyte population to assess whether there are changes within the live cells that are living within the tissue. And we found a variety of subclusters of the cardiomyocytes, but major finding was consistent within all subpopulations, which was that when we incorporate macrophages, cardiomyocytes downregulate, again, mitochondrial localized proteins within these live cardiomyocytes, including ATP5 citrate synthase, and they upregulate proteins related to glycolysis, including GLUT1 and MCT1. And so we're seeing here a shift from mitochondrial pathways to glycolytic pathways. And really, most of us might think that the opposite, that this might be a counterintuitive finding, in that if the tissue is functioning properly, why is it not relying on mitochondria? So we were interested in what actually happens in vivo in human development. So we went to publicly available single-cell RNA sequencing data of human fetal development. You can see it on the right side. And what we found is that when we looked at fetal cardiomyocytes from early development across many developmental windows, we see an overall downregulation of mitochondrial pathways. And so in reality, what's happening during development is a downregulation of these pathways and an upregulation of glycolytic pathways. And so what we see is that changes in cardiomyocytes within our microtissues induced by macrophages reflect the early changes that are observed in human fetal development. So I'd like to pose here some perspectives on these mitochondrial findings. And I'd like to show you here this paper that was published in 2020 in Cell that really showed that in a healthy mouse heart, so not in any setting of injury, cardiomyocytes, which are chock full of mitochondria, as we know, they eject their defective mitochondria into exopheres, which are then engulfed by macrophages. So this results in reduced accumulation of mitochondria inside cardiomyocytes, reduced accumulation of mitochondria in the heart, and overall, this results in improved function and metabolism. Now, in parallel to this, in my BioWire system, I observed that when I incorporated macrophages, I too saw reduced mitochondrial abundance within live cardiomyocytes, reduced accumulation of mitochondrial protein containing debris within the tissue, and this overall resulted in improved function and increased ATP content. And so this downregulation of mitochondrial proteins is similar to human fetal development, where we're favoring glycolytic pathways. And we know that this is largely due to this hypoxic environment, and shifting to glycolysis in human fetal development is actually required for the development of the heart and maturation. And so here we're showing that macrophages are a key part of this. Now, in line with these findings with macrophages reducing debris within the tissue, we also saw when we did pathway analysis that some of the pathways that go up with macrophages are those related to phagocytosis, or macrophages eating other cells. And so here we decided to look at a different time window within our system, and we wondered whether macrophages have a role in early tissue development. And so we focused on the first week of microtissue development and formation. And what we saw during this time is that in tissues with macrophages, there is reduced release of markers that are related to cytotoxicity or cell death, including LDH and cardiomyocyte-specific cell death, CKMB. We also see reduced mitochondrial DNA release, which we know could be either due to cell death or mitochondrial dysfunction, as well as reduced reactive oxygen species. And so at this point, we posed a hypothesis, and our question that we asked moving forward was, does macrophage uptake of dying or stressed cardiomyocytes result in reduced cytotoxicity and improved cardiac function? And so to assess whether macrophages are even eating cardiomyocytes within the tissue, we labeled our cardiomyocytes with a green dye called CFSC prior to making the microtissues. And then 3 days later, we looked within the tissue using flow cytometry, and we actually saw that within macrophages, we saw this CFSC signal derived from cardiomyocytes, which tells us the macrophages are uptaking cardiomyocytes within biowires. Now, I'd like to focus on a specific component of the cell membrane called phosphatidylserine, and this component is exposed on the surface of apoptotic or stressed cells. And so macrophages recognize phosphatidylserine on apoptotic cardiomyocytes, and they undergo efferocytosis, which is the specific phagocytosis of apoptotic cells. Now, we can block the recognition of phosphatidylserine using what's called a annexin. Annexin will bind to phosphatidylserine, and so we pre-treated cardiomyocytes with annexin prior to seeding biowires to prevent macrophages from eating. Now, to confirm, we can see using flow cytometry that when we looked at macrophages, we see reduced cardiomyocyte-derived signal in those that were treated with annexin, which together suggests that recognizing phosphatidylserine on cardiomyocytes is required for the macrophage efferocytosis of cardiomyocytes in biowires. Now, more interestingly, when we stop macrophages from eating, we see increased LDH, or increased cytotoxicity, as well as reduced contractile force and reduced relaxation, which tells us that macrophage efferocytosis was responsible for the reduced cytotoxicity and increased cardiac function that we see in our microtissues. So to summarize, we have these developing cardiac microtissues, and on the top are the control tissues without macrophages, where there's lots of cell death and the release of mitochondrial DNA and other toxic metabolites. But when we incorporate macrophages, they're able to eat dying and stressed cells, and this results in reduced cytotoxicity, improved cardiac function, as well as metabolic shifts that resemble those that are present during early human fetal development. And so to provide some applications here, We really think this may have some in vivo translational potential in cell-based therapies. So I introduced the challenges earlier on in my talk, challenges with cell-based therapy approaches, and we think macrophages can improve cardiomyocyte survival upon transplantation. And once transplantation occurs, we think that macrophages may help improve electromechanical properties, potentially preventing deadly arrhythmias that currently occur in preclinical models. We also believe that macrophages may be a key component in vitro bioengineering, not just in cardiac tissues, but also beyond. I'd like to thank members of a variety of labs. This was really a multidisciplinary effort. And I'd also like to thank MITO2i, who partnered with Ted Rogers to provide funding for this project last year. Thank you.
We have a time for one question for Humaira, if you have. Anna? You come back here.
Thank you so much. That was an excellent talk. I'm really curious whether, since you're studying in BioWire these embryonic stem cell derived cardiomyocytes, whether they're about the developmental stage, basically when you say that they're more glycolytic, and is this just this kind of phase that they have to undergo during development, and that is what you're really studying? Have you had a chance to examine what happens with the interaction between macrophages and cardiomyocytes in adult developed tissue and, a tissue that was injured, for example, and now wants to heal?
So that's a really good question. So stem cell derived cardiomyocytes, it's a major challenge in the field that we know that they resemble fetal cardiomyocytes, and getting them to resemble adult we're nowhere near that. There have been many papers just coming out in the last couple years, including those from Dr. Gordon Keller's lab, showing that we're able to do metabolic maturation, and they might, slightly go one developmental stage up. But if anything, we may be close to neonates with the most advanced protocols. But this is really, we can't study adult cardiomyocytes yet. And so that's why my study kind of now focuses on how macrophages interplay in development, which is still an important question because while we know a lot about what macrophages do in adulthood, we actually don't know anything about what they do in development, both in mouse and in human studies. And of course, in human studies, this is the only way that we can study development. And in terms of the glycolytic pathways, so initially we were struck by the finding that all these mitochondrial pathways were going down, and to tell you the truth, I spent 6 months just trying to figure out that problem. And so, but when we went into the literature, of course, we saw, this is actually what's happening in human fetal development, and the heart cannot develop without going through that glycolytic phase. And so that it was really amazing that that's what we saw in our tissues as well. And I think it's because if they're already in a hypoxic environment, and so relying on mitochondrial pathways, it's probably just detrimental to them. And they're probably just picking a pathway, the pathway of least resistance. And so, being able to move forward and study adult studies in the future is something we look forward to. But for now, this is really the systems that we have at hand, but it allows us to ask more interesting, novel questions about macrophages in development.